In this module, we will talk about different inducers that is needed in specific fermentations. Just keep in mind when we talk about the range of fermentation, especially the enzyme production. Enzymes are the biological catalyst and mostly produced through fermentation. Those microorganisms which produce the enzymes, and enzymes are basically the regulators, the biological catalyst that produce inside the cell, that control, and then ultimately that help in the metabolic pathways of any organism. So when we talk about the production of the enzymes through fermentation process, there are two kinds of enzymes. At that time, we classified into two kinds of enzymes. One are the constitutive enzymes, and other one that is the type of inducive enzyme. So constitutive enzymes are those enzymes which have to produce in all cases, because that enzymes are needed in primary metabolic pathways, without which the life of that cell is not possible. But as concerned, the inducive enzymes basically are those enzymes that is only produced in term of or in the presence of any inducer. So in those fermentation in which the enzymes has to produce in the response of any ingredient or any substance present in the medium, or sometimes they are produced when their desired ingredient or a carbon source or a nitrogen source is not present. So that is mostly we deal about the enzyme fermentation. So when we say that as an example of the starch is the inducer to produce the enzymes which we called as amylase. So mostly the mechanism behind the amylase formation as an inducive enzyme. Why? So that's why, because most of the organism mostly require monosaccharides or disaccharide as likely substances, more selective carbon. But when the media is lack of such monosaccharide like glucose, maltose, etc., and then there is the only carbon compound present in the medium, that is starch. So how starch cannot be directly absorbed by the cell or utilized by the cell. So first of all, then the organism has to switch on the central dogma which produce the amylase enzyme, and after the production of the amylase enzyme, either that is already present in the cellular body that excreted out of the cell, that will hydrolyze the starch into the maltose and or into then other derivatives, then that organism utilize. So the presence of the starch in the medium and the absence of the monosaccharide is the inducer. So that basically induce the production of amylase enzyme inside the medium. So there are so many examples with respect to different enzymes. Then another example is the presence of the cellulose for the production of cellulase enzymes. So in case of the enzyme nitrilase, so you can see here in this slide the nitrilase and the isovalerianonitrile that induce the production of this enzyme. So this substrate is basically the analog that are not attacked by the enzymes, may also serve as the enzyme inducer. So here in the next slide you can see the examples of various inducers. In the first case, as concerned alpha amylase, starch or the maltose used by Aspergillus and Bacillus subtilis in case of bacterial fermentation. In case of pluronase, that is the enzyme of amylase family, so the presence of the maltose. As concerned the alpha manosidase, then yeast mannans. So penicillin ecylase, and the inducer is phenylacetic acid. In case of the protease, then there are the various proteins. So according to the nature of the protein present in the medium, then that can induce to the production of that specific proteases, because we know that there are the number of proteases. So I have already told you an example of cellulase, and that is only produced in the presence of cellulose. Mostly the cellulases are produced by the organism commercially, that is Trichoderma viride. And then is the pectinases, that is only produced in the presence of pectin. The nitrilase, as we have already discussed, that is produced in the presence of an inducer, isovalerone nitrolide. So this is Rhodococcus rhodocrocus. So these are the different enzymes and their inducers, and used by different organisms on industrial scale. So as we have already discussed that yeast mannan in streptomycin production is also used. So these are other examples. So you can see here that monocytostreptomycin has only the 20% of the biological activity of streptomycin. So in case of, you can say that the production organism when is the Streptomyces griseus can be induced by yeast mannan to produce beta manosidase. We have already discussed this inducer in the previous table. So most of the inducers which is included in the list, that is only for the microbial enzyme when we are dealing with the natural system and those microorganisms which are naturally involved in the production of different enzymes, which I told you that is inducive enzymes.